Thanks again for the time today. And so I'm going to talk today about how I think we need to rethink how we're approaching low back pain. And so for those of you that care for patients with low back pain, which is probably the vast majority of you, since it is such a ubiquitous problem, how we need to take the learning that we've had in the past and combine it in novel ways so that we can actually move the field forward. And I hope that more than give you some information today, I hope that what really I'll do is provoke some new directions for you to be thinking about things, because I think what we really need is more people working on this very complex and complicated problem. So just starting with my disclosures, which are essentially related to research funding, government funding, as well as some foundation funding, as you see here. And our objectives for today, first will be to look at the risk benefit of current biologic approaches for disc degeneration, how mechanical loading impacts biology and responses in the disc, and then finally to appreciate really the complex nature of both biomarkers and behaviors, which is quite complex in influencing outcomes for patients with low back pain. So why has progress been underwhelming? And I say this as a clinician and scientist in this space because I do think progress has been underwhelming. This low back pain affects about 90% of people at some point in their life. Despite that, our costs and interventions continue to rise despite the fact that our outcomes are not improving. So there's something missing here. And I think that, as I'll explain to you today, I think a lot of this is related to us losing the forest for the trees, if you will. And so what do I mean by that? Well, there's a lot of trees in the process of low back pain. So if you look at what happens to the spine, just with the accumulation of wear and tear changes over time as we age, it really affects every structure of the spine. Interestingly, each of these structures can serve as pain generators and do in some instances. So as we see the disc start to show changes with collapse and degeneration, the bone changes, facet joint changes, ligamentous changes, muscular changes, in addition to the behavioral, biomechanical, and biologic and inflammatory changes that we see, the question becomes, what's our target? What's our treatment target here? And how do we actually move the needle? And how, more importantly, do we define with the patient in front of us what should be the target of our treatment when all of these different pathologies can actually be contributing to and driving the experience of low back pain, which in and of itself is essentially a syndrome rather than a diagnosis. Well, what is the most common diagnosis? Most common diagnosis in low back pain is disc degeneration. Starts in your 30s. We start to see wear and tear changes of the intervertebral disc. We start to see loss of hydration of the nucleus pulposus, thereby loss of its shock absorbing capacity. We start to see changes in the lamellae of the annulus fibrosus with tears, which can lead to obviously herniation and other problems in the disc. And we see changes in the end plate, sclerosis, which impacts the ability of the disc to have nutrition access the central portions of the disc and further the degenerative cascade. The challenge is, just because this is the most commonly identifiable feature, does that mean it's the best thing for us to target? And we're going to talk a little bit about that and why I think that some of our targeted approaches related to the intervertebral disc have not moved the needle in terms of care. And I say this in all admitting that our laboratory spent probably a decade trying to identify novel treatments for disc degeneration. And I'll tell you why I think that path is still somewhat challenged. So let's talk a little bit about the biology of the disc. So what do we want to do if we're thinking about addressing disc degeneration from a biologic approach? Well, what we would need to do first and foremost is intervene prior to end stage anatomic disease. Once that disc is collapsed, it is unlikely that any biologic approach is going to restore the full size of that disc. And then we also need to make sure we're addressing the underlying pathology, not just the pain, which is the presenting symptom when patients come in. So what do we need to do? We need to shift this balance from one of catabolic activity to one more favorable for synthetic activity to try to rebuild the structure of that disc and do so before the horse is out of the barn, if you will. So gene therapy has been an area that many investigators have explored, our group included. And really gene therapy, the idea behind gene therapy is to restore that advantageous anabolic catabolic balance, but with a sustained effect. Rather than a growth factor injection or a single intervention, the idea with gene therapy is to try to do this in a way that, because disc degeneration is a chronic degenerative process, you need a sustained effect. So a couple different approaches that have been thought about for the disc. One is in vivo, where you actually can inject a viral vector loaded with your gene of interest into the intervertebral disc. And by doing so, get the host cells to produce your anabolic or anti-catabolic gene of interest. Another approach is the ex vivo approach, where you can transduce cells in culture with your viral vector loaded with your gene of interest, with then the goal of reintroducing those cells back into the intervertebral disc. Obviously, there's risk benefits of both approaches, both in terms of the efficiency of the effect, as well as the number of procedures, numbers of interventions, et cetera, but both have been examined. So here's some data from animal studies where we looked at a gene therapy approach for intervertebral disc degeneration. And what we did here was, so these are just mid-sagittal MRI images, in this case of a rabbit model of disc degeneration, where you can see the nice bright white intervertebral discs on these T2 weighted MRI images, and you can see nice disc height. When we do an annular puncture to the intervertebral disc, what we see is a slow, sustained degenerative cascade that we've characterized biomechanically and histologically, and from a gene expression perspective, to very much mimic the course of disc degeneration in humans. So you can see that loss of that T2 weighted signal, loss of disc height in these punctured discs. If in that same disc degeneration model, we inject the discs with adeno-associated virus loaded with BMP2, an anabolic molecule that helps to facilitate cartilage anabolism, what we see is relative preservation of that T2 weighted disc signal, as well as the intervertebral disc height. Similarly, if we take our adeno-associated virus and loaded with TIMP1, which is tissue inhibitor of metalloproteases, an anti-catabolic factor, we also see relative preservation of disc height, as well as T2 weighted signal, indicating preservation of the proteoglycan, the main molecule that imbibes water in the intervertebral disc. So this looks great. We look at the biomechanics of those discs, because not only do they have to look good on imaging, but they have to function well. And if we just look at creep curves, which is what you see here, after applying axial load to that disc, what I hope you can appreciate here is that there are some differences whether we load the discs with an anabolic molecule or an anti-catabolic molecule. And in fact, those loaded with BMP2 are stiffer than those loaded with TIMP1. Nevertheless, we do see some restoration of disc biomechanics as well, which suggests this is a promising intervention. We also can look at what's happening from a turnover perspective by looking at released fragments of collagen 2. So this is a marker CTX2, which is the C-telopeptide of collagen 2 that is released during remodeling. And we can pick this up in the periphery, in the bloodstream. And so we can measure this over time, even without sacrificing the animals. And what we can see is normally over time in animals that undergo a puncture, you see continued release of collagen 2 fragments into the periphery, which is what you can see in the red lines there. But both interventions, both with the anti-catabolic molecule and the anabolic molecule, show blunting of that release of collagen 2, demonstrating that from a biologic perspective, we're also having a beneficial effect. So, these things look great. So these work were published back in 2012, almost 10 years ago now. And so the question is, why has this not been translated broadly into human trials? And the punchline is safety-related issues. And so unlike other areas where gene therapy has made some progress, in the intervertebral disc space, there are significant safety concerns related with gene therapy approaches. And what we've seen in other examples is that even if we think we have a perfect placement of the gene therapy vector, if you have super therapeutic doses, or you have placement of the injection in the in the intradural space in particular, you can see fibrosis, inflammation, which can lead to paralysis or paresthesias in the animals and significant morbidity. So it's not without risk and a significant concern in a chronic process like disc degeneration. So what about stem cells? Stem cells have had a lot of attention placed on them. Why not think about stem cells to restore the intervertebral disc? And we did some work in this space as well, using that same animal model of disc degeneration. And these were stem cells isolated from, they were umbilical derived stem cells. And so they were placed in a hydrogel carrier or just in buffer. And what you can see here is again, relative preservation when in the discs that were injected with the umbilical derived stem cells in the hydrogel carrier. And again, promising from an animal model perspective. Why has this not been translated? Well, very similarly, there's a lot of safety concerns. If you have stem cell leakage, stem cells differentiate depending on their environment, of course. And while in the intervertebral disc, they differentiate nicely into chondrocytic-like cells, which can be advantageous for the disc. When they leak out into the epidural space, for example, they can differentiate into bone. And bone formation is not a good thing in and around the neural foramen or the central canal, and can be quite problematic. And we've demonstrated that in the animal studies as well. So in thinking about improving that, can we do something to keep the cells in place? And this was a series of experiments that were done by a small army of orthopedic residents who worked in the lab with us for a couple of years and looked at a novel delivery mechanism by which we could use a fiber optic light to go in and polymerize a hydrogel carrier in situ within the intervertebral disc in hopes of keeping those stem cells in place. And again, looked very promising. You could see, again, relative preservation of disc height, disc signal, area, even biomechanics of that disc. Nevertheless, despite this, we did see inflammatory changes and differentiation into bone with significant osteophyte formation in that epidural space, which is obviously not advantageous. So despite best efforts, there's still a lot of safety concerns related to these biologics. And so when we rethink, we think about that, why are these things not making it to prime time? This is largely the reason. And kind of the take home here is, there's been a lot of thought about natural and autologous approaches in the intervertebral disc. The intervertebral disc is a very unique environment. It is relatively immune protected. It is relatively avascular. And so it's a very unique space. And so just because we're thinking about using autologous stem cells or others doesn't necessarily mean it's safe. This was a study that we did years ago that really hit that home to me, looking at glucosamine. And so this came up just really in clinical work, patients saying, should we be thinking about glucosamine for back pain and the cartilage degeneration associated with back pain? So this was a study done by Ludine Jacobs, who was an orthopedics resident that spent some time with us. And what she did was literally fed the animals that underwent the intervertebral stab disc degeneration model oral glucosamine and followed them over a 20-week period. And what she saw was quite surprising. And we thought maybe the lab numbers were flip-flopped or something. And that is the animals that were fed glucosamine actually had worse degeneration than those that were not. So, assuming that just because it's natural or a supplement or, something autologous, that it would not cause harm is clearly not the case in the intervertebral disc, given its unique environment and biology. So what about, of course, the disc does not live in isolation. It lives under constant mechanical load. And so how do we have to think about the interplay between the biomechanics of the spine and the biology of the spine? And this is a very important consideration when we're thinking about trying to alter the biology. So I like to think about, because I'm a physiatrist, I like to think about mechanobiology as perhaps the safest biologic. And if we could harness the beneficial effects of mechanical loading to actually facilitate a beneficial biologic response, that could be very advantageous if we were able to understand that. So if you think about how the disc experiences mechanical load, it transmits that load through the extracellular matrix and cell adhesion molecules to result in signaling cascade changes that we can then measure in the laboratory. And so we've done this in a number of different ways. We've looked at tensile stress, we've looked at compressive loading on intervertebral disc cells to see what is happening from a biologic mechanism standpoint in response to these loads that would normally be experienced by the intervertebral disc. So here's some examples of those data, just to help you understand that the effects occur at the level of gene expression, so mRNA expression. So these are data that show relative gene expression in intervertebral disc cells isolated from the nucleus pulposus, the most center portion of the disc. And what we can see is, depending on the magnitude of compressive load, you can see anti-inflammatory effects at the gene expression level. So this first panel here shows you decrease in molecules like iNOS or MMP3 or COX-2 in response to mechanical loading. And in fact, suggesting that some modest levels of mechanical loading can actually be anti-inflammatory. What's interesting is the time effect. So if you take that same load that's anti-inflammatory, but you extend it out for a prolonged period of time, in this case, 24 hours, which is what's shown in the second panel here, you now start to see things being pro-inflammatory. So, maybe at the most basic level, this helps us to understand why sustained compressive load sitting at your desk all day, for example, could have a negative effect on the intervertebral disc itself, on the tissue itself. Similarly, we looked at tensile strain, and we looked at this in the annulus fibrosis cells that primarily experience tensile loading. And what we saw was very similar, that under conditions of inflammation, which is what's shown in the white bar, if we applied tensile strain to the intervertebral disc cells, we saw a decrease in the amount of gene expression of inflammatory mediators or matrix metalloproteases responsible for catabolism of the disc. And so this, again, demonstrated some anti-inflammatory effect of controlled levels of mechanical stretch. But again, duration seemed to be the most important variable. You could fatigue that effect. So if you take that beneficial tensile load, but you do it for a sustained period of time, you start to see a reversal of that anti-inflammatory effect. What's even more complicated is if we look at cells from the degenerated disc. So if we look at phenotypically degenerative cells and how they experience mechanical load, it's quite different. So unlike normal cells, phenotypically normal cells, degenerative cells have an enhanced response to inflammatory stimuli, loss of that beneficial response to anti-inflammatory mechanical load, and enhancement of their response to traumatic levels of stress. Excuse me. So what this really tells us is we need to be thinking about how we can titrate, if you will, different types of mechanical loading based on where that person is biologically or structurally with their disc, which gets to the idea of needing to personalize this. And we're going to talk a bit about how we may be able to start to think about personalizing this based on people's individual biology and biomechanics. So could we combine these? Could we think about combining regenerative approaches that I showed you previously with mechanical approaches? And I like to think the idea of regenerative rehabilitation, which is a term that has really garnered a lot of interest, and Fabi Ambrosio here at University of Pittsburgh, I'm so sorry, has done a lot of work in this space. This idea that synergy between the biology and the biomechanics can actually have an even further enhanced mechanical effect. So that there are favorable levels of loading, unfavorable levels of loading, and combining those with biologic therapies may give us the best effect. But in order to be able to titrate that, in order to be able to understand in the human, not in the animal model or in the cell culture dish, how we can identify that and titrate that is one of the key considerations. So how would we do that? Well, we need some type of peripheral marker. We can't biopsy the disc. We don't yet have sophisticated enough imaging capabilities to do molecular imaging on a regular basis in the disc. So we need some sort of peripheral marker to do this, to be able to translate this. And this is some work that was done by a very talented graduate student and postdoc in our lab who created a bioreactor for a spine segment, bone, disc, bone, could keep it bioactive, and then apply different levels of mechanical loading, axial compression, repetitive flexion extension, torsion, and measure what happens to the biology. Importantly, what he found is in addition to what happens to the tissue biology, he could also find released biomarkers into the surrounding media. And in this case, this shows you an example of where he measured a marker called CS846, which is a marker of proteoglycan turnover that changed in response to loading of that intervertebral disc and bone, that functional spine unit, if you will. And so this took us in the direction of thinking, well, perhaps we can consider finding molecular biomarkers that could help us in targeting treatments. Not only is it a needle in a haystack, it's a bit more like a needle in a needle stack. It's really difficult to sift these things out and identify where the biomarkers are coming from in order to allow us to use these as potential targets. Nevertheless, it is showing some signal, and I'll show you some evidence of that. So this is a study that we did looking at older adults with axial low back pain and peripheral circulating biomarkers. So we drew blood from these patients before and after physical activity, where they were doing some walking, some repetitive axial torsion, some sit to stand activities. And we looked at the change in those molecular circulating biomarkers to see, was there any difference that seemed to associate with their experience? And we found many biomarkers that did. I'll give you an example of a few that are particularly interesting. One is RANTES, which is a systemic marker of inflammation. And what we found, and these pie charts just show you the R-squared value, so the relative predictive contribution to the observation. So RANTES had a significant association with patients' gait speed. It also had a significant association with their performance on a short performance physical battery. And so this was interesting to us to see that a marker of inflammation was changing in response to physical activity and actually correlated with their performance in these functional tasks. We also looked at neuropeptide Y, which is a marker that a circulating neurotransmitter that has been associated with resilience in patients with chronic pain. And what we saw, interestingly enough, was that the change in NPY seemed to be associated with these patients' response, emotional response, if you will, things like the affective score on the McGill, which asks questions like, is your pain punishing or cruel? Fear avoidance behaviors, so their fear of movement, as well as depression scores. And so, there was some differentiation between these two biomarkers as to these patients' experiences of pain and what seemed to be primary drivers for them in terms of their overall pain syndrome. We also noticed that these two different biomarkers, again, associating with different aspects of their pain at baseline. So the NPY at baseline was associated with, again, the McGill affective, the depression, and the fear avoidance, while as RANTES seem to associate more with functional capacity. And while you might say, well, these are really modest and unimpressive R-squared values, which I would agree with, this is a small cohort of subjects. And I would compare, when we compare that to the association with what is quote unquote our gold standard for diagnosis in low back pain, which is the MRI, where we have a little more enthusiasm about those modest associations, because the association in these patients with their MRI scores, whether we look at that based on a computerized index that is calculated based on T2 weighted signal and size of the disc, or whether we look at that based on the radiographic read, the radiologist read of those in a scoring system, we see absolutely no correlation with their pain. This shows their pain, but we also looked for correlations of their MRI findings with their function and other markers, and saw essentially no correlations. So it suggests that there's something to look at there, and I'll tell you a little more about what we're doing now to further hash that out. But of course, go ahead.
Question. Since you invited us to ask questions in the middle, I will ask now. But so those biomarkers that you were, is this in chronic? I mean, it's very interesting that you have a stronger association with the mental health markers than other things. So I'm wondering, is this in chronic patients, and in the context of when are you making the assay of these molecular biomarkers? Are they doing an exercise and then you withdraw blood, or are they doing a baseline? And is such a big distance between? I don't know.
Yes, it's a great question. So this cohort was all older adults, 60 and older, with chronic axial back pain. And what we did was we just brought them into the lab. So it wasn't a certain period of time after the onset of their symptoms. They had to be defined as chronic. But we assayed them when they first arrived to the lab. And then we did a series of physical activity maneuvers and assayed them after that. And we were looking at that change. And then we were also looking at the association between some of these metrics and their baseline value. Now, your question is important because baseline is going to be different in everyone. It's going to depend on their diet, their medications, their medical comorbidities, all of these things. And we do see quite a bit of variability in just those baseline levels that are completely completely unrelated to their chronic back pain. And so sometimes looking at this delta or change in response to physical activity can be more helpful, if you will, than looking at the baseline. But if you think about it from a pragmatic standpoint, what we really need is something to give us a trigger on what we should be doing. So we need something that's going to give us information about where that patient is at that moment, which is why we're starting to become more interested in genotyping. And I'll talk a bit about that. But yes, which is why I alluded to it as a needle in a needle stack, because baseline levels are affected by so many different things, and with these circulating markers, most definitely. Yeah. So to that point, here's an example of where we looked at this at the genotype level. So in hopes of trying to address the issue of when to assay, how to assay, other contributors. So because we were interested in neuropeptide Y, and for lots of reasons, one, it's actually surprisingly produced by the intervertebral disc, even though it's a neurotransmitter. That was somewhat unexpected to us. We did look at this, and the disc itself can be a source of neuropeptide Y, which is in and of itself interesting. But we were interested in this marker and wondered if we could see some genetic variation. So we looked at known single nucleotide polymorphism in the NPY gene. And we looked at this in a population of patients with spinal stenosis and symptomatic spinal stenosis. And we subdivided these patients as responders or non-responders to the intervention. So this was a piggyback to a randomized trial that was looking at different treatments for spinal stenosis. And what we saw was there was a difference based on the genotype in the responders and the non-responders, and who carried the SNP and who did not. What was even more interesting is when we looked at the 3 different intervention groups within the clinical trial. So there were 3 interventions. They were randomized to either usual medical care, which essentially included medications and epidural steroid injections, and or group exercise, just a group exercise program, or standardized physical therapy. And what was interesting is in the patients that underwent the usual medical care, there was no difference in their response rate based on genotype. But in both the group exercise and the physical therapy, there was a difference between responders and non-responders based on genotype. So, again, this is just a potential signal in many, many signals that could be explaining this, but one that has garnered a lot of interest. So, but can we take this one step further? It's one thing to look at, whoops, my slides are stuck, there we go. It's one thing to look at this in a cohort, but it's quite another to think about it potentially guiding treatment. So we did a study looking, and we asked, well, let's think about something episodic, because it's hard to say responder, non-responder in chronic low back pain, because it's this chronic disease that you have to look over a long period of time and a lot of patients. But something episodic, like response to an epidural steroid injection. Could we look at this in that cohort and see if there are biomarkers that might help us figure out who should and should not undergo an injection? We did this in axial low back pain, and you might say, well, why are you doing epidurals in axial low back pain? Anyhow, the evidence isn't that great for it. But what we see over and over in the literature and over in clinical practice is there are a lot of injections that are going on for axial low back pain. There seem to be a cohort that do respond to it. We just don't know who they are a priori, which means when you look at this over the entire cohort, you see a pretty poor treatment effect. So is there a cohort within there that actually has the potential to be treated? And can we identify them a priori to then only inject that group? And so we recruited patients that were adults that had low back pain worse than any other pain in their body to try to limit that contribution from other areas. And we're having a spinal injection performed as part of their routine care. We purposely did not influence whether or not they would be receiving an injection. We approached them for consent only after they consented to undergo an injection. So we had a relatively small cohort, 48 subjects that underwent a lumbar epidural steroid injection. And we defined responders as 50% or greater improvement in their numeric pain rating scale. So a pretty high bar. Usually we think about clinically significant improvement as much smaller than that, but we kept a pretty high bar. And we ended up with 17 responders and 31 non-responders based on those criteria. And importantly, we also looked at those patients' disability index, and those that we classified as responders also had a clinically meaningful change in their US-Westry disability index as well. So what we saw is that there were genetic biomarkers that, again, were different. That NPY marker that we were using previously was different in responders and non-responders, as was the vasopressin receptor, which has been looked at in chronic pain populations as well. But even more impressive than that was the difference in catecholamine methyltransferase, obviously involved in the adrenergic system. And what we saw was pretty specific that, that non-responders or those carrying the A allele in the SNP were all non-responders. So it was a fairly dramatic, even though this is a relatively small cohort to hang your hat on, it was suggestive that maybe, this might be a direction that we could go to use this as a predictive tool. So how can we put this all together? And so one of the things we're thinking about now, and this is really just to get you thinking about how could we use these. So what we very much don't think is that there's going to be a magic biomarker. There's going to be one marker that is going to be suggestive or predictive of patients' response to treatment, particularly in something as complex as low back pain. But can we start to combine large data sets in ways that give us better predictive capabilities? And I'll give you just a bit of a teaser on that. So this was that same cohort of patients that underwent epidurals. And if we look at just their clinical data, we looked at their profile based on their medical comorbidities, medications, et cetera, and created these phenotypes of patients who were responders and non-responders, just based on their clinical data. And that's what's shown here, the red being non-responders, the blue being responders. We did the same thing with the genetic data, the single nucleotide polymorphism data. And again, we saw some that were clearly defined as responders, some that were clearly defined as non-responders, but a lot of overlap of those peaks, right? So a lot of uncertainty still in that analysis. But if we took the clinical data and the single nucleotide polymorphism data and we put them together and did that same phenotypic analysis, we saw much better separation of those cohorts. So much better ability to predict who was going to be a responder and a non-responder. And this is really the direction we would like to take this next. And I'll tell you a bit about how we're doing that now. But just suffice it to say that, because this is a syndrome and because it is multifactorial, putting all of those factors into the analysis is likely going to lead us down the road of a better predictive capability. But one thing we can't ignore is the behavioral component of this, which is so important and so substantial in chronic low back pain. So I'll touch just briefly on these items because this is something that we need to incorporate into our clinical decision-making. So there are a lot of barriers that patients have to their recovery. So outside of the structure of the spine, outside of the inflammation they're experiencing, there are a lot of things that are impacting, limiting, and complicating patients' recovery. Things like their mobility itself, pain that prevents them from reactivating and becoming active, fear of movement, anxiety and depression, which are so common in this patient population and can create a vicious cycle, and lots of medical comorbidities that commonly coexist. So what we've done clinically is we brought, like many others have, we brought these practitioners together in single clinics in order to try to tease out these different components and remove some of the barriers that patients have to recovery. And this just gives you a sense of how we have thought about that here in Pittsburgh, what we have titled our musculoskeletal health program, where patients are managed by a nurse care coordinator. We collect patient-reported outcomes. Most of these patients are quarterbacked by a physiatrist. They're always seen by a physical therapist in their initial assessment. And then we connect them with some of these other resources. We have a dietitian, we have a pain psychologist, rheumatology, sleep medicine, anesthesiology, all working together to try to coordinate and provide the comprehensive care for these patients. And so what has this looked like from an outcomes perspective, despite the fact that we get really the most complex patient population. So the vast majority of our patients are classified as medium high risk based on StartBack tool, which is a common assessment for risk for response. They've done very well. And so probably the biggest win is almost 70% of them are actually converting into going to physical therapy and reactivating and becoming more active. And a quarter to a third of them have seen significant improvements in their functional scores, their disability scores, with relatively low utilization. So this is why these programs work, because you're actually removing some of the unnecessary utilization and replacing it with some utilization that's actually going to move the needle on removing barriers for long-term recovery. This works nicely in our environment because we are an integrated delivery and finance system, which means we own our own insurance company. So about 40% of the patients that we serve are self-insured, which means the money we dump into these programs is realized as a benefit to the insurance side of the house and can be reinvested in the program. And so that helps to keep things keep things to be sustainable. And so how can we then think about further individualizing the care that we provide for these patients? Obviously, not everyone needs a dietitian. Obviously, not everyone needs a spinal injection. Not everyone needs a psychologist. How do we hit that sweet spot to make sure we're giving the right treatment for the right patient? And so we really need to refine our decision-making capabilities. This gives you an example of one of the dashboards that we've created to do just that. And this is, again, this example is looking at epidural steroid injection. Again, we kind of use that as one of our test cases because it's episodic and it's easy to measure. But here's an example of where we use just the clinical data to create 10 different phenotypes of patients who are going to respond or not respond. And then we can use this. This is actually a visual on the dashboard that the clinician can sit down and look at this with the patient and say, okay, here's your individual likelihood based on our clinical prediction of your likelihood to respond to an injection, which is what's shown in the green, or we're actually worsen from an injection, and do a shared decision-making with that patient. This is an example of a patient who has a moderate degree of disability, but really no comorbidities. If instead we look at a patient who is taking a muscle relaxant, an opioid, a nonsteroidal, an antidepressant, they have fibromyalgia and IBS, you can see how dramatically that changes their likelihood to respond to that injection. And again, this can be used in a shared decision-making way. And then finally, if you take a patient just like that and you layer on anxiety and depression, you then see a much higher odds of not responding to that injection. And so we've used this, and we've done some quality improvement initiatives looking at what are the outcomes, and our patients choosing to still move forward with the injection. Interestingly enough, some do, and we've followed that over time. But this has been a very useful tool for our clinicians in trying to sort this out and individualize those treatment decisions. So in the end, what we need to do is the right treatment for the right patient at the right time. And I'll leave you with this idea of how I think we can get there. And it really relates to big data. So what I've shown you are small signals in small data sets. And in order to really get this to the next level, we need lots of data, comprehensive data, and large data sets to do this. And so here's an example of that where we were able to use big data. So we have within our system access to over 500,000 adults with low back pain. And so we can mine those data sets and start to develop more robust phenotyping capabilities. And this is an example where we looked at that cohort of subjects, and we were particularly interested in the question of the patient's likelihood to continue taking an opioid for their back pain versus their likelihood of discontinuing an opioid for their back pain as a surrogate marker for things going in the right direction, if you will. And what we were able to create is all these distinct phenotypes. And so all these different colors represent a different phenotype for a patient and their own likelihood to either remain on that opioid or come off of that opioid. And again, we can map an individual patient onto this based on their individual characteristics. But what we can also do is mine the phenotype itself. And so look for the interconnectivity of the different variables within that phenotype to give us information about what else we should be looking for. If we just keep looking for treating the anxiety and the depression, or we keep looking for treating the inflammation, or we keep going after the same variables, we're likely missing some important pieces. And so this is an example of how we can do that, where we take one of those phenotypes and we say, what are the characteristics that make up that phenotype, and what are the associations within each variable? And we see some interesting nodes that we can then start to develop some hypotheses around. So in this case, we saw in one of our cohorts that a very interconnected node was whether or not they saw their primary care physician. So what do we need to do with intervention at the primary care level to start to have an impact on these patients' trajectories? And so it helps us to look at some things that we might not be thinking about doing. So that's exactly what we're doing with our newly funded center. So we were fortunate enough to receive a very significant grant from the NIH to fund a mechanistic research center in low back pain. This is part of the Backpack Initiative, which falls under the NIH HEAL initiative. And what we are doing as part of, this funded center is we are recruiting 1,000 subjects with chronic low back pain, and we are doing deep phenotyping. So pretty much everything I showed you today, we're going to look at in all these patients and then some. We're going to biosample. Well, we are biosampling these patients. We're collecting saliva, serum, and spinal tissues for anyone that moves forward into going into surgery. We're doing proteomics, genomics, transcriptomics. We're doing deep phenotyping of them from a biomechanical perspective, where we're looking at them in the lab based on biplanar fluorography, where we can look at segmental biomechanics. We're sending them home with wearable motion sensors for their spine, as well as accelerometers to look at general activity and look what's happening in the field. We're doing assessments of them from a physical function perspective in the laboratory. And then we are collecting a very significant number of patient-reported outcomes, social determinants of health, and lifestyle factors that we think that if we start to look at this and then combine all of those things. I showed you that brief example where we just combined clinical metrics and single nucleotide polymorphisms and got better predictive capabilities. Our hope is from a machine learning approach, if we take all of these literally tens to hundreds of thousands of data points and start to put them together to define better phenotypes, that will lead us to the next step of not only directing care more appropriately, but also figuring out how do we develop novel approaches to care in this patient population. So this is a really exciting project, and maybe if Pablo invites me back in a few years, I can tell you what we've learned. Hopefully we will move the needle on this, but point being, it definitely takes a village to do this. And so we have people from bioinformatics, from physical therapy, from PM&R, from psychology, chiropractics, engineers, molecular biologists, all working together to try to put everything about this syndrome together and hopefully better define where we're going. So it's very daunting. I am always painfully glass half full. And so I think that we will be able to eventually figure this out in a more targeted way so we can improve treatments. But with that, it takes interdisciplinary teams. I'm speaking to the choir here. You guys know how to do that very well already, but it also makes for a very fun work environment to bring people from very distinct backgrounds together around this common problem. And I'll just end by mentioning one of the things that we're doing is starting to put clinicians and researchers together. So this is an exciting new initiative, and that construction site pictures is now a little data. We actually have 4 floors built on our new vision and rehabilitation tower, which is going to pair researchers and clinicians literally in the same space, bouncing off one another, so that we can try to continue to move things forward more quickly. And it's interesting because we are combining ophthalmology and rehabilitation. You might think, well, that seems a bit distinct, but if you think about visual impairments, cognitive impairments, and mobility impairments, and how people negotiate their environment, there's so much synergy and overlap that we're finding that it's been a very, very exciting project. So I'll end there by just thanking you for the time and your attention today, and certainly more than excited about hearing your thoughts or suggestions or questions if we have a few minutes.
Gwen, thank you so much for this fantastic talk. I really appreciate the chronological perspective of where we are today on this story. So this is great. I let people ask questions maybe while they're unmuting. I'm gonna ask maybe the first or the second now. But so this HEAL project, the backpack project, this sounds awesome. It is in the context of standard of care medicine. So patients are going through whatever the physicians or other providers are suggesting to the patients to do.
Yes.
How you take that into consideration? Is this also, put this as a variable in machine learning and hopefully something comes out?
Yeah, it's a great question. So this is an observational cohort. We are not in any way directing treatment. We are not in any way influencing treatment. And what we are hoping to acquire is patients who go through a lot of different treatments, right? And that is what we're seeing so far. So we're about 220 subjects into our 1,000 cohort. And what we're seeing is, with the exception of we're actually seeing very few surgical patients, which is interesting, and we'll have to figure that out, but lots of different treatments. And we have a very detailed questionnaire and we pull this from the electronic medical record too, that outlines their treatments, the timing of their treatments, when those treatments are in comparison to our assessments as well. And yeah, my hope is that dumping this into that machine learning approach will help us say, okay, in the cohort that underwent physical therapy, in the cohort that underwent an injection, in the cohort that had medication management, how do we sort that out? But we are part of a larger consortium. So there are 3 mechanistic research centers, one at UCSF and one at Michigan. And we're also going to all dump our data in together and look at that. The consortium is currently planning a randomized clinical trial that will start in 2022 that will also be doing all this deep phenotyping, but now we're going to direct the treatments. And so we're going to have a 4-arm study. It's going to be a smart design where there will be some crossover. And hope to be able to help tease some of this out that way as well. But this group at this stage, because we don't know the phenotypes yet, is purely an observational cohort. Yes.
Great, thanks. Eric?
Yeah, Gwen, yeah, great talk. I was really curious. I was interested that you mentioned this sort of tool that lives in the PM&R system that, sort of has all these different data points that, sort of give this odds ratio about whether the patient's going to, be successful for treatment, things like that. Could you just talk a little bit about, I mean, it seems like you guys are collecting a lot of data. You mentioned, depression, information, this and that, and how are you guys, how are you guys, what's been any, advice, suggestions in terms of systematic collection of that so that you guys can kind of do that in clinical care? You sort of mentioned, this is maybe a quality improvement project, but can you speak to that a little bit? Because I think sometimes, having, providers collecting lots of data can be very challenging. And so I just was wondering how you guys have been successful with that.
Yeah, it's a great question. And that example that I showed was very little PRO-based data. It was more pulling data out of the, diagnoses, medication prescriptions, those sorts of things, rather than PROs. Because you're right, the kind of systematic collection of patient-reported outcomes is challenging in its consistency, and it's challenging in its implementation on a broad scale. So our clinical analytics, while we have the PROs and we put them into, we use Epic here. And so we have them in discrete fields in Epic when we're collecting them. Of course, they're not being collected all the time. We're moving toward promise measures that we will have much more robust collection because they'll be collected in the primary care setting, in the specialties office, all of those sorts of things, not these kind of one-off episodic, which is what we've had to deal with in the past. But we have focused those explorations more on the standard fields in the EHR than we have on the PROs, with the exception of, for that example, we used Oswestry Disability Index, which in our system had been collected on subjects, whether or not they were in PM&R's office or PT's office or surgeon's office, et cetera. So we were able to use that. But we've tried to mine the data that is standardly accessible and not rely as much on those things that we rely on the patient to collect for the reasons that you outlined. And because we have big numbers, we're able to do some of that. Now, with our cohorting, with our research projects, we collect like 45 minutes, of questionnaires where patients fill out almost, it takes them almost an hour to fill out their questionnaires. We can't do that in the clinic, obviously. So we need to pick and choose. And so that's why we're going to focus more on some promise measures that hopefully will be a little more streamlined. So, yeah.
All right. Thanks so much.
May I ask a question?
Please, Jeff. Thanks.
This is not going to be of so much general interest, but I wonder about having so many different data points going in. And is there something that you do to reduce the likelihood that you're going to find type 1, type 2 errors just on account of there being so much data?
Yeah, it's a great question. So, are we going to send ourselves on a wild goose chase for the next decade because something pops up by chance that we decide to chase down? It's a great question. And one I wrestle with all of our bioinformatics colleagues on, our machine learning folks and AI folks. And that's why we're going to do both. So we're going to take multiple approaches from a machine learning perspective, but we're also going to take standard statistical approaches in testing some of those hypotheses within our cohort. And what we were actually just talking about earlier today was just on with our NIH program officer. And we were talking about using each other's large data sets within Backpack to validate some of those. So we'll develop these. And what I think is going to come out of our data set are hypotheses that need to be validated on other existing data sets. And that's what we've exactly discussed doing, is swapping big data sets. So we'll have this massive database of, a couple 100,000 data points, we'll develop our clinical predictive ideas, our hypotheses essentially, and then hopefully we'll swap with UCSF and say, let's test them on this other cohort of subjects and see if we're able to validate it. But I think, so my short answer is we're going to need to take multiple approaches so that we don't go down a rabbit hole. And then we're clearly going to need to validate this on other data sets. And then obviously down the road, we'll need to definitively test this prospectively to see if it's actually meaningful in patients, or have we just, found the red herring? So it's a beautiful question.
Beautiful answer. Thank you.
Gwen?
Hi, Barbara.
Hi. This isn't scientific. I just wanted to say I noted a nice touch where on many of the references you included a photo of the young author.
Yeah, thank you. So we really, it's one of, for me, the most rewarding part of all this work is seeing some of our scientists in training start to accomplish this. So thanks for noticing that. Yeah, we like that.
Great. Well, I think we are a few minutes after the hour. Gwen, thank you so much for joining us today and really sharing this. This is really nice work. So hopefully people get inspired and maybe want to connect and follow up with you and so on.
I'd love it. I'd love it.
It's awesome.
You have lots of data to mine. So
Yep, absolutely. Great. Thank you again. And thank you, everybody. Enjoy the rest of the day.
Take care.
Bye.